Hello, it's second year students. Now we are going to start with the fungal cultures one by one. Okay. So first of all, we will try to see the lower fungi and their structure, their somatic part, their spore structure and everything. So our lower fungi is having coenocytic hyphae, which is having more amount of nuclei in the hypha, but it will not have any suppression or differentiation of the fungal cell. Now at the tip of this pharyngophore, there will be a structure which is globose in the shape, and this particular structure will contain the asexual spores which are called sporangiospores. Entire structure which is sac-like is completely filled by the spores like this. Okay. The spores will be dirty white in color. This fungi gives us cottony, fluffy, filamentous growth. It is found to carry out infection and growth in the vegetations, okay, in the fruits, in vegetables, and also it is found in the soil. Now this is called sporangiophore. Sporangiophore is the structure which is holding the reproduction structure, that is reproductive part. Okay. This is called coenocytic mycelia. It is called substrate mycelia also. Now this is considered in the category of aerial mycelia because it is found in the air. This is our sporangium. And the spores which are located inside are called sporangiospores because they are found inside the sporangium structure. So I think you are very clear with all the parts of the mucor. Okay. So because it is coenocytic in nature, it does not have any special structure. It can carry out a sexual reproduction by formation of sporangiospores inside the sporangium. It is also having the holding structure, okay, that is called sporangiophore, and all the hyphae it has is coenocytic in nature. Okay. Now we will see this is our mucor. Okay. So there are different species of mucor, mucor racemosus, mucor roxy, okay, and so on. Now we will see the another structure of fungi and another genus of the fungi, that is Rhizopus. Rhizopus is also the fungi which is found to be related with the deterioration in the bread chapatis and all other vegetations will have the growth of this particular organism. When they grow, the color of the spore will become darkened, that is go to black side, and that is why when they mature, when the colony get matured, after long incubation, it will look like dark blackening okay, of the spore. So that type of thing is found. First of all, we will see the structure of this organism when you do the microscopy. Okay, this way you will see the sporangiophore. Okay, then this is our coenocytic hyphae. This is our coenocytic mycelia, which is already having many nuclei in its structure. Okay, and directly opposite to the sporangiophore structure, there will be a roof-like structure. which is called by the name rhizoid. And because it is having these rhizoids, it is called rhizopus. Okay. Now at the top of this sporangiopore, there will be a globose structure which is having very big size compared. Okay, comparatively, it is double than the size of the sporangium of the mucor. Okay, which is usually found, and it is also having some branches. So like this, and here in this bigger sporangium, comparatively bigger sized spores are there which get blackened upon maturation of the fungi, and in the entire structure of this sporangium the spores are rotated and complete sporangium is packed with the spores. Okay, so this is our rhizopus genus. Okay, so now we will call this as sporangium. These are our sporangiospores. Okay, these are our rhizoids, and fungi is considered as the lower fungi. Okay, it will also give you filamentous growth. Now we'll switch over to the next genus, and that is Aspergillus. That is Aspergillus. Now in case of Aspergillus, it is considered as higher fungi. It is considered to be part of Butyромycotina. And now we will draw the structure of the Aspergillus when we see under microscope how it will look like. Okay. So here also the same thing is there. Choanocytic mycelia is there. Then you can say conidiophore is there. This is our conidiophore. It is called conidiophore because here we are going to see the conidiospores on the tip of it, and that is why it is called conidiophore. This is our choanocytic mycelia. Still it is choanocytic because this is considered as the intermediate or between lower fungi and higher fungi. Okay. Now this is our conidiophore, and at the tip of the conidiophore there will be a structure called vesicle. This is called vesicle. And on the tip of the vesicle, on the periphery of the vesicle, there will be a chain of stigmata. These all are stigmata. You can see that in all the directions they are located on the vesicle like this. And on the tip of the stigmata, now the spores or spore chain of the spores are found. It all depends on the species of the Aspergillus, that how these spores are arranged in the chains. Okay, so there are also different types of the chains on depending on the species of the Aspergillus genus. Now these organisms give you granular growth or little bit velvety growth. It will not give you cottony fluffy filamentous growth. These fungi produces certain these type of spores which are colored. So usually they give yellow colored spore or blue-green colored spore and also black-brown colored spore. So three different type of colors are observed in these fungi. Okay, it may be black, brown, or it may be blue-green, or it may be yellow also. Okay, so there are different type of species of Aspergillus, and depending on that you will see the coloration in this fungi. Okay. Now on the tip of this stigmata there is chain of spores. Okay, you can also make the chain like this. Okay, on all the stigmata, and this is called conidiospore. This is called conidiospore. Okay, and this fungi is called Aspergillus. Aspergillus is involved in producing so many diseases in animals and human beings. It is also found as a contaminant. It is also behaving as a saprophytic organism. It is found in soil, in vegetation, and in decaying material. Okay, fine. Now this organism is having different species like Aspergillus flavus, Aspergillus nidulans, Aspergillus niger, and so on. So this is considered as the higher fungi. Okay. Now yes, I just want to tell you another point which I am missing. At the opposite end, okay, at the base of this particular conidiophore, there will be a special structure in the Aspergillus which is called by the name foot cell. Foot cell is only found in the cultures of Aspergillus and it is not found in any other cultures. Okay. So this is the identification criteria of these fungi, and it also tells us that this is the intermediate in between lower fungi and higher fungi, because from this particular fungi the evolution starts in the fungi and septation has now already started here. Okay. Because you can see one single individual cell in the phagocytic mycelia, okay, at the part from where conidiophore is emerging from the mycelia. Okay. So this is considered as the special structure of fungi. It is only found in Aspergillus genus and it is considered as the foot cell. Okay. Fine. Now we will switch over to the next higher fungi, and that is Penicillium. Now in Penicillium, this is the fungi which gives you a velvety appearance of the colony, and it is having the coloration like blue-green color. Okay, the spores will give you blue-green color. It is found in decaying vegetation, in decaying fruits. It is sometimes found in the refrigerated materials, and this organism is frequently found in the agricultural plants. It is also able to produce certain diseases of animals and living beings. Now these organisms are considered as the higher fungi, and being higher fungi, they will give you septation. Regular septation is found in each and every hyphae and mycelia in this organism, and you can see that the aerial mycelia will turn itself, or it is terminated in the form of a special spore bearing structure, and that is called conidiophore. This is called conidiophore. This is our conidiophore, and this is our septate mycelia. This is septate mycelia. Regular septation is found from Penicillium onwards. We are going to see so many other cultures. Now at the tip of these there will be again steric in the odd numbers. Okay. Okay, that will be 3, 5, 7, 9 and so on, depending on the species of the Penicillium. Okay. So here there will be chain of the conidium or conidiospores like this. This culture is a free steric. Okay. Fine. Now sometimes that is also possible that maculae is found. Okay. First of all, maculae are there, and then steric will be found on the top of the maculae, and then conidiospores are located in this view. So it all depends on the species of the Penicillium. Okay, how it will look like. This is called palm structure. Okay, this fungi is involved in production of antibiotic, and that is called Penicillium. Okay. So this is about the higher fungi. Okay. Now rest of the fungal cultures we will also going to study now. Okay.